welcome to the colloquium. My name's Claire Yu, and I have the pleasure of introducing this colloquium speaker for today. I think most of us with COVID, we're very glad to now be back in person. But one of the things about COVID and the pandemic was that it raised our awareness of pandemics and pathogens and the importance of our immune system. And some people who had slightly defective immune systems had a much worse reaction to COVID than others. So immunology and the immune system is really a fascinating field, and it's only recent years, I think, the physicists have begun to get involved in immunology and the immune system, and today's speaker is certainly one of the experts in moving in this direction. So Armita Nourmohammad is associate professor physics at the University of Washington in Seattle. She also holds appointments in the Department of Applied Mathematics and Computer Science and Engineering. Works at the interface of statistical physics, and in particular non-equilibrium statistical mechanics and biology. And she uses that to develop theoretical data-driven approaches to the evolutionary processes across scales. She earned her PhD in 2012 from the University of Cologne, and then went to Princeton University, where she was a James McDonald postdoc fellow. And that's where she started, I think, working on immunological problems from a biophysics and evolutionary perspective. And I think some of that she's going to be telling us about some work on in that area. So in 2017, she started splitting her wave function between Germany, where she was a group leader at Max Planck, and the University of Washington, where she joined the faculty. And my understanding is the research groups got quite entangled, somehow got a little more localized with COVID. She has numerous awards and honors, including the NSF Career Award, the NIH Mira Award, and she received the APS Division of Biological Physics Early Career Award, which is one of the reasons why when I tried to organize a symposium, we needed people who did physics and immunology. We wanted her, but she was disqualified because she'd already given her talk for the that award. So anyway, it's a pleasure to welcome Armita. Please join me in welcoming her to the stage.
Thanks, Claire, for a very kind introduction, and thank you for coming to the colloquium. My fellow biophysics program attendees put a slight pressure on me to represent the field. So if you like the talk, please generalize. If you don't, you can blame it on me. But what I'm going to tell you about today is more recent endeavor in my group trying to, again, get at the immune system and how immune system interacts with pathogens, but now using ideas or tools to study proteins and how that factors in immune recognition. Right. So from COVID, what we know is that we know the good actors in the battle against pathogens, right, are immune cells usually. Bad guys, if things don't go well, get sick. My group is really interested in understanding how in different types of interaction between the immune system and our body would lead to different outcomes of an infection. Just to give you a very sort of brief overview of how immune system works. Our first line of defense is called the innate immune system. It's basically anything that is non-specific, including your skin barrier that keeps the pathogens from getting in. But once infection sets in, we have our adaptive immune system. We and the rest of vertebrates have the adaptive immune system. That is a very fascinating machinery to specifically battle infections. So the main actors in our active immune system are B cells and T cells. And we have a very diverse repertoire of these cells. So they express this type of receptors on their cell surface. And we have tens of billions of these different receptors in our body. So each cell has its own receptor effectively. So because of this diversity of the receptor repertoire that we have, we can counter specifically diversity of pathogens deal with, right? So you have one receptor against a fragment of some pathogen. That's why you get vaccinated, you produce new receptors, antibodies. So, the T cell receptor, when it gets secreted in the blood, we call it antibodies. So you sort of learn how to specifically target pathogens. Just a little bit about B cells and T cells. This is a B cell with its receptors. Generally, B cells see the outside of a pathogen, right? So it's bacteria or virus that you have, they interact with proteins on the outside of the pathogen. T cells, on the other hand, they see the inside. What happens is a little bit more sophisticated, right? So you have a T cell. Generally, all our cells in our body, we express these other receptors on our cell surfaces called MHC. And these receptors, it present short fragments of proteins that are inside our cells. Most of the time, when the cell is healthy, these fragments of proteins are from your own body, right? So they're self. You're not going to mount a response against them. But if you have an infected, if you have an infected cells, so you have proteins from the pathogen also in the cell, and they get presented on the cell surface. And once that happens, a T cell can recognize that and go and battle that cell, kill the infected cell, then recruit other types of cells to go after the infection. So T cells see the outside, T cells see the inside. Together, basically, they cover the whole pathogen. Now, this video that you're going to see is you'll see a T cell trying to go and basically kill an infected cell for years. And so if you follow them, you see basically the infected cell gets moved from the body or killed. It happens of the order of 10 minutes, 20 minutes once things are in contact with each other. Generally, if you want to look at the time scales relevant for this process, once things are in contact, it's about 10 minutes for a T cell, killer T cell, to kill infected cell. The cells are immune cells communicate with each other to recruit each other to sort of form an army to go against an infection. That's of the order of an hour or so. They differentiate into different roles, so they become killer, effector cells, plasma, become memory for future infections. So that happens of the order of 5 or 6 hours. They proliferate into larger clones to really start the army, right? So that's of the order of a few days. So that's when you start actually feeling sick. You're producing a lot of these immune cells during that time. And if you then go at the sort of scales, right, at the beginning, you're really looking at molecular recognition. And then as you go further, you have populations of cells that are going after the infection. So you start from molecular scale to a cellular scale. And there's also a further time scale that is of order of months and years. So if you think about the immune system at the population level, so all the humans on the planet, trying to deal with pathogens that we are seeing around us, think about COVID, right? Every few months, there's, depending on what the type of pathogen, new variant comes up. And that's basically escaping the collective immunity that the population has built up over with these processes. So as a result, you get a co-evolutionary process that happens on the order of months to years, depending on the type of virus you're looking at. So in my group, we are interested in all of these time scales and sort of end scales, if you wish. But I'm not going to talk about all of this today. But yeah, so these are really the scales that are relevant in the process, and they interact with each other. So that's the sort of unique thing about biology. Separation of scales, not really a thing. And so we need to think about how things go from very small scales to large scales. Right. A little bit more about recognition, in this case, T cell recognition. So I mentioned that we have these receptors on our cell surface that present whatever is in the cell, so just surveillance of whatever is cell, and T cells go around to recognize them. In this case scenario, what you want is if you're recognizing an infected cell, right, a sort of red fragment of a pathogen, you want to respond really fast, of the order of seconds, to actually deal with that infected cell. But generally, we show a lot of self on our cell surface as well, and characteristic time for that response should be infinity. You don't want to go kill that cell, right? So there is this time scale that you need to respond fast to an infected cell when you recognize it. Right? also need to be very sensitive. So generally, even at the peak of infection, when cells are presenting these protein fragments on their cell surface, most of the things you're seeing are cells. So you have the pool of about 1 million cells, you have about 10 fragments from a pathogen. So the amount of proteins you have from humans in a cell is much larger than what you get from a virus. So this is a difficult discrimination problem, right? So you need to take a 10 out of 1 million and do it fast. So you need to be very sensitive. That's a difficult problem. On top of that, what makes it even more difficult is that the difference between self and non-self is not huge. So if you think about, molecular dissociation, that is sort of a proxy for affinity of these interactions. Itself would basically fall off. If you bind to a self, a receptor binding to a self falls off about a few seconds to 3 seconds. Non-self, 5 to 10 seconds. The affinity difference is not huge. If you think about using equilibrium mechanisms and the difference in the sort of concentration of these peptides or fragments and the small difference in their affinity, it's really hard to use equilibrium binding to discriminate an infected cell from non-infected cell. Let it be in a fast manner. So what the cells do, well, they spend energy to do that, right? So they use non-equilibrium processes to do that, and specifically, they use sort of proofreading or kinetic proofreading that has been introduced previously by John Hopfield and others to actually go after these problems. As a result, by pumping in energy and using that, you can have a fast, sensitive, and a specific response to infection. So that's a fascinating thing. A lot of physicists have been studying these processes and sort of building upon them. And I just wanted to sort of bring this to your attention because some of you may be interested in sort of looking into this process. Now, did I hear? Right. So what we had so far was a diverse repertoire, a specific, sensitive, and very fast response. Your experience probably look at here. All right. You know that our immune system also develops in our own lifetime, right? So if you see a new pathogen, you mount a new response, you get ready against it. So before COVID was around, we didn't know anything about it, but once you get infected or vaccinated, now you have the right immune receptors, antibodies, and T-cell receptors to go after it. So it's the sensory mechanism that develops throughout our lifetime. It learns about the environment and it adapts accordingly. It's really like a secondary nervous system in your body that is dealing only with pathogens. It also keeps a memory of infections, act more readily in future encounters. So in many ways, it's really similar to nervous system, but it does different things. And the cool thing about it, it's kind of, once you say it, it's obvious, but it recognizes pathogens that did not exist when you were born, with being an example. So this is a system I'm really fascinated by, and I've been studying it in many different ways. What today I want to tell you about is trying to think about how the space of these immune receptors are organized to counter the pathogens that we see in our environment. So what you're seeing here is sort of a abstract representation of this space, called shape space, if you wish. So on top, it was what we would call immune shape space. So here are your immune receptors, and they can be close to each other. And if an immune receptor is close to another, that means that they can recognize similar pathogens, similar types of antigens. And then there's a dual space of what we call antigenic shape space. So that's the part that pathogens live in. And so if there are two pathogens that are close to each other, the non-self pathogens that are close to each other in this space, it means that they can be recognized by similar antibodies or T-cell receptors. Now, these are beautiful pictures. Physicists really like it. We build models based on this, but we have no clue at the molecular level how this thing is organized. This was one of the questions that I'm very interested in. Can we say something about molecular structure of this space? And we learn that from data. If you pay attention, there are also parts of this space that are identify as self. So this is your own self protein, and the idea is your immune system should not mount response against that. So the sort of also hypothesis was that there are holes in the immune shape space that basically are associated with where the self would be located. I haven't really seen those holes, I can say, but that's something to look for. Now, there are different ways of looking at this. We have looked at this looking at a lot of sequence data that come out of these receptors. We can do modeling to study this system. But over years, what I've realized is that to really understand this immune shape space, you need to think about these receptors as what they are, which are proteins. Here, what I'm showing you is an antibody or a B-cell receptor protein at the bottom interacting with the antigen protein on the top. And so up here is a T-cell receptor interacting with that receptor that's supposed to be expressed on your own cell, it's called MHC. It presents a little peptide from antigen, this line here. And so we're dealing large with protein-protein interaction when we're thinking about immune pathogen interaction. Now, B cells can interact with other types of molecules like lipids and sugars and things like that. Most of the antigens we deal with are some sort of proteins. So to understand immune shape space, we need to understand something about protein shape space that is much more general. And the hope was understanding that would tell us something about our immune system. So before getting into this, I'd like to acknowledge the people that have been working with me on this problem. So most of the work I will talk about today has been led by started by a graduate student, Mike Poon, graduated now over a year ago, and continued by others in the group. So the work I'm presenting will be Mike Poon's and John Marco-Visani's work, and collaboration with Phil Bradley, Jakub Atrunovsky, and Paul Thomas over the past few years. And it's a recent sort of direction in the lab. Basically, we started it during COVID, and somehow, Mike, one graduate student at the time, he wanted to do this, and somehow it grew in scale because we got excited about it. All right, so let me tell you some basics of proteins. You may have heard about amino acids, right? So these are the building blocks of proteins. And so generally, especially as theories developed in biophysics, we think of amino acids. There are 20 letters. You just write them down and just deal with them as letters. In reality, they're molecules, right? They have atoms. And all amino acids, they form this type of backbone that is common among them. And they have what's called side chains. And the side chains, these different colors, are the things that are different between one amino acid and another. We have 20 typical amino acids that our proteins use to form their structures. First step in the process to form a protein is you go and have an amino acid sequence. So this is a sequence of letters, these 20 letters after each other. So the primary structure that can fold spontaneously to a tertiary structure, a 3D structure of a protein. For a typical human protein, that takes about milliseconds in our body to happen. And then once you have a structure, that determines the function of your protein. So what other types of protein it interacts with. So you have protein-protein interaction, what channels it goes through in your body, and things like that. So there's this paradigm of protein sequence to structure to function map. I'll sort of re-emphasize that as we go forward. Now, there's been a lot of work trying to study different parts of this map. You may have heard about AlphaFold. That's asking the protein folding problem, mapping from a sequence to a structure. So that's been a breakthrough, basically, in this field. Now, I think it's a few years ago, using really machine learning engineering to get at these problems. A lot of people have been working there over 50 years, right? So there's been a lot of development in the field, and then AlphaFold really built on those developments to have a really usable tool that is successful for many proteins. Now, other maps that people are really interested in is going from sequence to function. And the models, especially in these days, machine learning, are under the umbrella of these large language models. So these are ChatGPT, but now for proteins. So instead of training it on text and Reddit and Shakespeare and so on, train it on protein sequences. And so that gives you a lot of power to go from sequence to function. I can't say the problem, but the thing that both of these maps have to deal with is they have to go from things that are long-range interaction in protein sequence to learn something about local interactions in structure. Because your sequence, once it folds, things that are far away in sequence, they can come very close to each other. So that's the part that forms biophysical interaction or real physical interaction. So your models usually have a lot of long-range interaction information in them. And that makes them actually hard to interpret because it's a many-body interactive system. On top of that, these are just large black box machine learning approaches. So what we were interested in was basically having a slightly more interpretable model and thinking of if we start off with the structure, do a structure to function map. If we do that, you're dealing with local interactions in the structure, and we may be able to actually learn more interpretable models in the system. So the type of questions we would be interested in asking in that case would be, if you give me a protein structure, can you predict binding affinity of the two structures? Can you design better binders by changing some of the amino acids? These are concrete problems you can pose. They happen to be difficult to answer. So how can we address that? Now, structure function maps are attractive, but there's a challenge, and that's the data imbalance. The attractiveness of sequence models is that there's a lot of protein sequences. So if you go to a protein database these days, about 10 to the 8 or more protein sequences deposited in these databases. You can build really good machine learning models for them. You go to, yeah.
An equation, f of a bunch of variables equals E.
Yeah.
E, so like the energy.
So say binding energy, if that's something. And the variables in that case was coordinates of your atoms and identity of your atoms in this.
That's some figure of merit, lower the.
Yeah, pure. Can replace it by anything, any other function you care about. So if that was a binding energy, binding affinity that you care about, that's the thing you're trying to. So the data imbalance here is that we have a lot of sequences, far less protein structures. So there's a database for protein structures, but we have 100,000 of those. You can add computational structures with AlphaFold. You get a few million, right? Still orders of magnitude smaller than the sequences. If you care about, say, antibody structures or T-cell receptors, it's far less. So about 1,000 antibodies and hundreds of T-cell receptors. So it's really hard to construct any statistical model or data-driven model in this case. Well, how do we go about that? So the idea that we had was, if we start from a structure model, we don't need to model the whole structure, but rather we can learn local models of these structures. Because if it's the biophysical interactions and you learn something about it here, it might apply to other places in the proteins as well. It might apply to other types of proteins as well. So we can have local model, and it can relate to, if you build it in a way, learn effective physical interatomic interactions in your model, then it can be generalizable. So you might just need your data to go by. And the hope is, if you infer such a model, you can infer function, and you can generalize to, say, B-cell receptors or T-cell receptors without having to really train on extensive amount of data in both. Okay. So if you inject physics in your models, you might be able to do something. Now, the way we approach the problem, this local problem was, let's say I start from protein structure like this. I want to learn statistics of the microenvironments in this structure. So let's look at one amino acid here in the protein structure. So this is what's highlighted. Now, what I want to do is I want to mask this amino acid and then look at the atomic composition around this amino acid and ask the identity of the one that is in the center, right? So this is a simple problem, right? Well defined. Why is it interesting? It's interesting because it's effectively going to learn some effective potential. Given the composition of atoms around you, it's going to learn effective potential for that amino acid in the center. We think of this as some building block of larger protein interfaces that then, once you have the model, you can put them together and build a larger protein interface.
Very clear the solvent around the protein.
yeah, so that's something we are interested in. It's hard because some of the protein structures, not all the solvent is crystallized with it, right? So you get some of the water with it, and it's frozen, but not everything. It's hard to actually solvate the protein algorithm.
Sorry, before you go on, I mean, I've seen these structures a million times.
Yeah.
I never understand. There's like spirals, things like that, and nobody ever explains it out.
Okay, what are they?
People tell me, oh, you need to look for like the cap beads, and that's where things can bind. But like, can you get the 1 minute explanation of like what we should be paying attention to?
So this is a cartoon of a structure. If you look at the real protein structure, right? So it's just point cloud of atoms, right? But then I mentioned at the beginning, amino acids lie on these backbones, and then they have side chains, right? So side chains are usually the sticks that you see coming out, right? These guys. Then backbones are things that they lie. So you can draw these cartoons using where the backbone lies, right? So and sometimes you form sheets, sometimes you form helices, sometimes you form loops. So these are the forms that they draw, right? But you're right, this is confusing, and I don't care about this shape. Basically, what I end up caring about is really atomic composition of the structure. So just points in 3D space. That's what I will go by.
So just to add to that question, I mean, when we look at this, so you're saying those sticky things are like side chains, and the ribbon is the backbone.
Basically.
It's like a three-dimensional representation.
Some representation.
Those components of this protein structure.
Exactly. But we don't really care about it, right? At the end, what I will show you as a representation in 3D is really points in the space. Atoms, where I put in, XYZ coordinates.
You can show us another representation of the protein.
Yeah.
That's more useful than...
That's what I use, right? Because I don't want to tell my algorithm what is backbone was. I mean, I didn't know when I started working on this either. So just think of this as points in the space, and these are just for, to guide your eyes where amino acids are lying. Right? So that's the only thing we can think of. All right. So the idea here is I give you this atomic environment where I mask the thing in the middle, right? The amino acid I care about. I don't know its identity. I don't know any of the atoms that are part of it. From all the other atoms around, I want to have some function that tells me what's the amino acid in the middle, right? That's a question. Now you can go build your physical model, right? Really write down some Leonard Jordan's potential and try to characterize this. It's going to be more principled, easy to interpret because you put it in. Our experience shows that doing that, it'll be usually very expensive computationally to compute these things. Alternative is machine learning. It's fast, powerful, but usually a black box, hard to understand what you've done. So we were asking the question, can we make this less black boxy, more interpretive? So to just give you a very brief overview of what we are thinking in this area, right? So you have, for deep learning or any machine learning, you have an input, you have an output. Say this is your atomic composition, this is the identity of amino acid, and you want to map them. So you want to learn some function that maps one to the other, right? And generally machine learning, right? So what you do is you approximate this by combination of linear and nonlinear functions. So these linear matrices, the W's, right? These have weights in them. When you're training a model, you're learning those weights. The phi's are nonlinear functions that you put in your algorithm, and it's fixed. And you can show that this is a universal approximator to whatever function you are trying to. These are learnable. These are what's called nonlinear activation. And having nonlinearity is actually what makes it expressive and work. Now, generally, you think about the space of possible maps, f of x's, right? Can be the true map of the problem, and there may be some degenerate maps in this space. Maybe there's a subset of maps that respect the type of physics you care about. So we are interested in this subset of.
Good question. So these nonlinear activations, or we don't also learn?
You put it in, right? So you know them. So it's not guessing. I mean, you have choices, right? So, but usually what you do is something that sigmoid-like or it's called the ReLU function, so it looks like something like this, right? And they're useful for various reasons, right? It's easy for optimization and very. You put many of them on, you stack many of them on top of each other, then you have a function that is expressive enough that gives you the right map. Yeah, so we're interested in physics-respecting maps. How do we get there, right? How do we respect physics? The first thing usually is when we are thinking about theory is what are the symmetries of our problem, right? Symmetry is a big deal in physics, right? Physical theory. And going back, when thinking about physics and symmetry and conservation laws, right? Noether's or Noether's theorem, right, tells us that when we have time translation symmetry, right, we have conservation of energy. And we have translational symmetry, conservation of momentum, and rotational symmetry end up having conservation of angular momentum. So these types of symmetries are relevant. We are writing down theory. More or less at the same time, Einstein, when he introduced the principle of covariance in the context of relativity, the idea there was that we're not the center of the universe, right? The laws of nature should have the same form independently of the reference frame you're in, right? I've been teaching electromagnetism recently, right? So we have the equivariance between electricity and magnetism, right? So when you're standing still with respect to a charge, you're going to measure electric field. When you're moving, right, you can measure both electricity and magnetism. And our theory basically allows us to go from one to another in a very well-defined way, right? And what we want to do, and goes for acceleration and gravity, right? What we want to do is have a theory that, or have a machine learning approach for proteins that respect the type of symmetries we care about in our data and follows this principle of covariance. How do we build that? What I do is I would replace the physics-respecting map here to a symmetry-respecting map. Doesn't guarantee that I get physics out of it. That's the hope, right? So that's the step because I know how to constrain symmetry easier than how to constrain physics.
Wait, so you have the same shape block, though. You think one is the sum of the other, or?
No, so probably not, right? Probably not. I was just lazy. But yeah, so different blob, but the hope is, one gives you the other. Okay, so what kind of symmetry we care about? So we were looking at there are various types of symmetry, but if you're looking at this neighborhood problem that you have an amino acid at the center, obvious symmetry is that if I rotate it globally, nothing should happen to the energy function that I want, right? So it should be rotationally invariant, right? So interatomic interactions should not depend on the orientation of your molecule. That's obvious symmetry. So say I have this molecule, I look at some part of it that binds to something. I'm trying to learn some binding energy to some other molecule, right? So there's a neural network. Now I have rotated version of the same molecule here. They all should give me the same energy. A conventional machine learning approach is, as humans, we know these are all the same protein, right? When you stare at them. The algorithm doesn't know. A conventional machine learning approach would basically train a model by rotating by training a model, but all the rotated version of the same protein. In 3 dimensions, you can estimate that you need 500 versions of these orientations to actually train a reasonable model, capture symmetry of the problem. So that gives you an invariant model. Now, this is a computationally extensive, but also it does not guarantee that you get the same model for all of these orientations. The worst case scenario is that you get one model for each orientation, that the output is always the same energy, but the structure of the model can be very different. Now, if you inject symmetry in your problem, right, so if it's explicit in your machine learning algorithm, symmetry of our neural network, you only need to train with one example in one orientation, and you should be able to generalize to all of them. Right? That's what we are trying to get at. Well, the goal is encode our data, so this protein structure data, in a rotationally symmetric way. then process data with our neural network in a rotationally symmetric way, then using that to learn function. So there's been a lot of development in this field trying to think about how to constrain neural networks to respect certain types of symmetries. So these goes under the umbrella of group equivariant neural networks or geometric deep learning, initiated by Max Lane and Michael Bronstein and others. So we started building on those sort of theoretical developments to apply those for our protein structure.
Yes. So in a lot of machine learning applications, if you're lacking data, you can use this rotation property, increase the amount of data you have to non-predicate some threshold. So do you have a problem with hitting a threshold? This only works like this shit data. I'm actually sort of surprised that you do.
I mean, once you're not building new data when you rotate data, right? You're just trying to teach the problem that, cat standing up is the same as cat rotated. Both of them are cats, right? So you're not learning a new animal in the model, right? So in the image processing, you're just telling it, this is a semantic and this is orientation and separated from each other. So you're not getting anything out of it in that sense. You're not getting any semantic information out of it. So we have enough, well, we'll see how much, if we have enough data. So in this case, we have, right? But by rotating your objects, you don't generate more semantically informative data. You just generate semantics from orientation.
By that argument, the learning threshold curve should shift. Once you get implemented, that you shouldn't need as much data.
Yes, you do not need as much data. That's it.
Question here. So I'm not sure I understand what the problem is, because chemists have to deal with this rotational translational thing all the time. They just use internal coordinates.
Yep.
Which is that.
You can do that, but so that would be invariant representation. So basically, we say I just look at the distances, relative orientation, right? You can do that. That's an easy way in. What we have seen and others have seen, if you put this as input to your algorithm, it won't be as expressive as the way I would explain to you, the way we are going to do. But that's basically one obvious way of doing it.
So one more question. If you've done the green thing, another rotationally symmetric encoding, why do I need the brown thing?
You give me a minute. Right? So I'll get to it. Depends how you encode it, right? So how do you want to operate in that space that you have encoded it? Okay, so let me just give you one concept that I'm going to use. So invariance is an obvious concept here, right? So let's say I have a water molecule. I'm measuring its atomic mass. If I rotate it with this operator dr1, atomic mass should not change. So atomic mass is invariant under rotation. So if this is the operator of rotation on the molecule, I get the identity operator on my measurement, right? Now, there's a concept, equivariance, which is a more generalized form of invariance. That's when I am measuring, say, a dipole moment. It rotates when I rotate my molecule. So now I have this other operator that lives in the space of dipole moments. It tells me how to rotate my dipole moment when my input is rotated, right? So if this is an operator that is well-defined, a linear operator in this case, right? We call this thing equivariant under rotation. So my dipole moment is equivariant under rotation. And internal coordinate, as you mentioned, right? So these are things that are invariant under rotation. What we care about is having a model that is equivariant under rotation, and we can go back to the invariance from them, and that proves to be more expressive. So how to encode data? When we have symmetry, usually we think about Fourier transform, and that's a useful way of sort of going about this. So the normal Fourier transform, say I have a signal, a spatial domain, right? So I can expand it in the Fourier, in the frequency domain, right? Using just the normal Fourier transform, minus kx. Now, if I transform my signal by some spacing a, I know my Fourier coefficients are gonna just transform by factor e to the ika. So this is now your dr2 operator that I mentioned in the previous slide. So this is equivariant under translation when I'm doing this Fourier transform. Now, if I have rotation, I need to do a slightly more sophisticated thing. Well, this is my point cloud in space, right? So it's a density. And I need to do sort of spherical Fourier transform. So instead of e to the minus ikx, I use spherical harmonics. Right? And then I get coefficients here that are now the Fourier coefficients, now in the spherical. Right? There is a additional coefficient here, n, because I have a radial component as well. So the YLM gives you the angular part, and then there is a radial component that takes care of that. And now what we know is that if I rotate the object, we know how our coefficients should change. They should change under the Wigner D matrix. Right? So we know how exactly that happens. Instead of e to the ika, I have a Wigner D matrix. So that's my PR2 operator. Right? Gives me the equivariance. This is a linear part of operation, right? So I just rotate things, but our machine learning algorithms also need nonlinearity. That's what makes them really expressive and powerful. So if I do the usual nonlinearity that I mentioned, the sigmoid function or ReLU or all of this, they break the equivariance. But again, going back to physics, we know how to combine spherical harmonics, how to combine spins, right? So that would be a Clebsch-Gordon tensor products, right? So you can get one spherical harmonic and tensor producted with another one, right? So that gives you effectively a bilinearity in the system that again lives in the space of spherical harmonic. Right? So this part is the very linear thing you do to your model in rotation. You know how to deal with it with Wigner D matrices. And these part basically gives you a power of adding nonlinearity or bilinearity in this case for neural network. Now the cool thing about it is the Wigner D matrices are also irreducible representation of the group SO3, which is the one what we are dealing with here, the rotation around a fixed point in the space. And so that makes it actually an attractive way of representing this data and dealing with this. Right. So these are the components of our neural network and our encoding that we're going to use for our problem. And the way we do it, now I'm going to go to a simpler representation of protein. So this is my protein structure. And these are the point clouds that I really actually work with. So this is a distribution of carbons of this structure in space. You have the same for oxygen, nitrogen, sulfur, all the other atoms, with some other properties like charge and salsa. But really, these are many different delta functions in the space that in three-dimensional space that you're dealing with. The first step is you encode that using a Fourier transform, and in this case we're going to use Wernicke transform, which has a specific form of radial function. It doesn't matter that much. The result is some superposition of three-dimensional holograms, spherical holograms. That's why we call this a holographic encoding. But this is really your input to your neural network. The raw data, so far nothing is applied to it. And then you train your model, which is now a 3D rotationally equivariant neural network. And the reason it is a special type of model is that you're now living in a Fourier space. And for example, your weight matrix usually combine different parts of your data with each other. You cannot combine different L's with each other because then you break your equivariance. You won't have the same Wigner D matrix applying on it when you rotate your data. So you'll have a constraint on what your weight matrix can be. So that's what makes that model, the brown part that we showed in the slide, to be rotationally symmetric, rotationally equivariant. So neural network you're using here should have a specific sort of rules in what coefficients are non-zero. And the way we train it is like you have all these steps, and at the end, you're going to get basically a bunch of numbers, 20 numbers associated with the 20 different amino acids. We call those numbers pseudo-energies. You run it through a sigmoid function that looks like a Fermi function. Then you get probabilities associated with these different amino acids at that point. That gives you what's the likelihood of having an amino acid at a given position in this model. Yep.
What defines the size of the nucleus?
I define it. So 10 angstrom. That's the size. You vary it and you see what works. And so 10 angstrom is a good number. And so because it's as small as 10 angstrom, we don't have anymore. We have millions of these neighborhoods. We collected from all the protein structures, right? So we have a little bit more data than the total number of structures that are out there. Now, but how do we do with the task that we set out to do? So this is what we call a confusion matrix. So these are the input amino acid, the 20 different types. The output, if you were perfect, you would lie on the diagonal. So we have an overall accuracy of 70%. I don't know why that pops up. That's the state of the art for this particular problem. And mistakes we are making actually make sense. So if you look at how the mistakes cluster amino acids with same physical and chemical properties and the size cluster together, I can replace one by another, and that's allowed. We know amino acid can mutate from one type to another, and that's basically what's captured in our model. So you can measure how much replaceable a specific amino acid is.
Just with an idea, what is the computational time you need?
Yeah.
Is it a minute, a month, a year?
Oh, 1 hour. Yeah.
Even if you repeat the question, because they don't.
Okay. So the question is, what's the computational cost of this? So the computational cost of this, the training takes 1 hour on a GPU, normal GPU, a single GPU in a computer. So reason, this was the next point that I wanted to make. The buggy, right? The peer, not. Right, so it's much faster than if you would train this as really trying to deal with images, right? Trying to interpret these point clouds in the image. It's actually 100 times faster than that. The reason we are thinking that's faster is because we are constrained to the symmetry of sort of symmetry-respecting maps in the space. We're not searching all maps, searching a much smaller subset of possible maps, and that makes it much faster, our optimization. And when it's faster, then you can do a lot more with it, right? So far, the task that we set out to do, we did the first part. Is it learn physics, right? So that was the second question. So very naively, you can think about, we're looking at crystals. They're frozen. There should be in some sort of minimum energy of some energy landscape. If I draw some energy as a function of physical distortion from the original crystal that you had, you should see the original protein to be at the minimum, and then every other distorted version should be sort of above, right? Higher energy. Now, can we use our network to get at this? The way we did this, we computationally distorted these proteins, and we did it by sort of twisting them again around the backbone at different positions. So what we are seeing is some examples of these twists. So this is on y-axis, you have the score in our neural network, which is a proxy for energy as a function of distortion. And what you're seeing is that the originals, the zero in distortion, the native structure, are usually at the energy minimum. So indicating that maybe our network has implicitly learned something about the physics or interatomic interactions that we have.
I'm not sure I understood that part. So your network, you put in some coordinates of all your things that gives you a profile of what it guesses the amino acid is. Now I put in distorted profiles of density of where oxygen and carbon are. I get a different profile of probabilities. How do I...
But you know what the amino acid is, right? So you want to know for that particular amino acid, how much the probability or log probability has changed, right? And that's the difference in energy. So that gives you really, that's what we interpret as different. So that seems to be something that, looks quadratic with a minimum at the right place. And it's not proof that it's learned physics, but it's proof that it's doing okay.
Thank you, Shem. Understand your training, but here, the training set is basically you take the protein structure, right? You hide one of the amino acids.
I take 10 angstrom within a protein structure around the amino acid of interest. So I take many different neighborhoods within one protein, and I do it for all protein. So the training data is about 1 million of these neighborhoods collected from many different proteins on PDD.
And if the test data the same setup.
It's the same setup. but we make sure the proteins in the test data has no evolutionary similarity to training data. Like, we really make sure these things are very different from each other.
But the goal is not to predict the structure, it's to actually understand the energy landscape research.
That's the thing that came out of it. That's not the goal. So the input is really protein structure, right? And if you wish, this is a mask language model, but in the space of a structure rather than space of sequence. You mask some center, you try to learn what should be in the center from the surrounding.
Yeah.
Yeah, I'm a little bit confused about whether it could have been any other way, because you were minimizing log likelihood during your training, and now you are saying that the minimum was at 0. What, isn't that just two sides of the same coin?
So the distorted structure could be in the data, right? So I'm minimizing over a huge set of data, right? So, and the distorted structure could appear in the data as well. So this is not on a specific protein that was, you're training on. Is something completely different. Yeah, I mean, that's a good point. Again, this is not a proof, that's physics, but we have more proofs and I can tell you later about those. But yeah. All right. The hope was now using this model to predict function and to do something that generalized to the proteins that I'm interested in, with T-cell receptors and B-cell receptors. Given the time, I'm not going to cover all of this. I'm going to hit at some points here. So let's look at the first one, the stability effect of mutations. We started with this protein called T4 lysozyme. It has many different mutants of it resolved. The structures of those are resolved. So the mutations, basically, you change one amino acid to another. So here, for example, pointing at 2 amino acids. Then if you go from orange to yellow, right? So that's a neutral mutation, so it doesn't change the stability of a protein. If you go from purple to pink, it's a destabilizing mutation. It reduces the stability of the protein. The nature is faster. And there are experimental measurements of many of these mutants for this particular protein with their corresponding protein structure. So that's a unique data. And so if you're looking at stability effect, is a change in the free energy starting from wild type, the original structure, minus the mutant. Well, delta G, because each of them is delta G. So for wild type, you look at the wild type protein structure. For mutant, you look at the mutant protein structure. Right, that is, in this case, 40 of them, different types of mutant. Each point in this plot, right, it is one original amino acid, so I at position 3 mutated to P. And this is my network's prediction on the wild type structure on this one, what the stability of this wild type amino acid or energy associated with that wild type amino acid. So I have 40 of them, so these are all the 40 colors, the different positions and different amino acids. Now for mutants, I look at the same quantity, but then for each of these, so P here, I will look at the structure that has P at position 3. Then I give it to my network, that neighborhood, and it gives me a number, a log probability, and just I plot that. So, and my assumption is that these two are proxies for my free energies, the wild type and the mutant. And so if I subtract them, I should get something like stability effect, delta G. And red is bad, blue is good. So if you qualitatively split, because we know what is destabilizing, what's neutral, you see actually the split is right. And so out of this 40, you can actually plot the stability effect, a prediction versus the true experimental stability effect. And we see about 80% correlation. This is, again, this is a model that is not trained to learn about the stability, but it picks it up because it picks up something about physics. So we have more examples of many more proteins and in a new preprint here, and models that are specifically refined to predict stability, and you can boost this performance by a lot, actually.
The delta G on the x-axis, these are measured experimentally?
Yeah. So you change temperature and you wait until the protein destabilizes.
Are you learning about function?
So, I mean, stability is somewhat function, but we learn about binding as well. I'm gonna skip this part because I want to go to T-cell receptors, but we can learn binding effect of mutations as well. So this is a binding affinity to different types of molecules, the small molecules that our model did not see at all because we only trained on proteins, so we do well on that. Now going to T-cell receptors, we were interested in modeling binding affinity of T-cell receptor with a peptide and MAC. Right? So the hope is if I have a complex, pick a neighborhood somewhere here, and I ask, what's the goodness of that amino acid at that position, that tells me something about the affinity of that interaction or the contribution of that amino acid in that affinity. We're interested in predicting affinity and designing new peptides. So the challenges here, again, as I mentioned, the affinity, the data is scarce, right? Data is highly degenerate, so you have many-to-many maps. You have also very low affinity of T-cell receptor. So it's a micromolar affinity, which is orders of magnitude lower than a typical good binder that you have in biology. So if things are low affinity, it's hard to predict them generally. The question is, why is this an interesting question? The answer goes to cancer immunotherapy. These days, we use T cells to battle cancer. So we know T cells can discriminate between self and non-self to some extent. But if you have mutations due to cancer, your self will be slightly mutated, right? And sometimes it's mutated enough that your T cell can recognize it. And the sort of questions in the field is that how well T cell receptors or TCRs can recognize these cancer antigens. So what's the limits of our repertoire in recognizing cancer? Can we design better T cell receptors to do that? If I design a good receptor, is it going to bind to also self? Is it going to be self-reactive? So how specific would it be? The question of specificity is really equivalent, like what's the space of possible peptides, these little antigens that a specific T-cell receptor can bind to. If it's very specific, so it recognizes one, so that's good. I can design it to deal with just mutated self, and I'm good. But if it's very broad, then if I target the mutated cancer antigen, it's going to attack your own cells as well, and that's not great. Right? And that's one of the issues with cancer immunotherapy. On the affinity prediction, so again, the data is very limited. We had few examples that we started with, and again, using our model without any further training. So these are different names of different peptides and T-cell receptors. So each of them had these many measurements. Our model actually predicts okay in certain cases, right? So 90% correlation, 50, 66. Again, nothing trained on this. But I think the most exciting part, let me skip this one as well, is trying to design these peptides. And the hope was, given that we have learned some sort of energy landscape, and we give a T-cell receptor, an MAC molecule, and maybe a little bit of peptide, try to design other peptides that could fit in that structure. So, sample from the energy landscape. The idea here is start from these. So, MAC, backbone of the peptide, so we don't know the identities of amino acids. Sample from the energy landscape, the side chains. You relax the structure, you iterate, and at a structure that works. So there was some original experimental data that was available in this specific system I'm showing you for this specific T-cell receptor and MAC. So the thing you're seeing here is a probability distribution. It's a very weird way of showing it. These are different amino acids at different positions, right? The colors tell you a little bit about, tell you about the chemical properties of these amino acids. So this is experimental data that they scan over various peptides. These are our predictions. They get the general sort of features right, but there's quite a bit of difference. And we were disappointed at the beginning, but then we gradually learned that these experiments were not so quantitative. This probability distribution should not be taken very seriously. But then I gave this talk, and Thomas, our collaborator, told us, oh, I can actually generate make these peptides in the lab and go measure them. And so we generate a bunch of peptides. Now, this is a different T-cell system. We've done a bunch of them. We designed 93 different peptides, and we asked, what do they do in the lab? Right? So this is a plot that walk you through. These are experimental activity that Aja here did measurements on. These are our predictions. We did 35 negative designs originally. We knew it was negative. We wanted to calibrate the model. And this is, I think, about 60 positive designs that what we did. So anything above 1 on the experimental side is active. So it's a functioning peptide. And so this guy is a false negative, right? So we have a single false negative. And then we have basically 50% accuracy in our design, which is when it comes to peptide or antigen design, usually you get far less than 10%. So this is quite successful in that area. And again, none of these models are trained on T-cell receptors. These are trained on general protein universe. And we learned rules of proteins to go and design things that didn't exist. So if you are interested how different the false positives and true positives are different from each other, you can look at the logos, the probability distributions. See, there are 2 positions that we didn't do quite well. We sort of had more variability here. Now, what we are doing is there are more experiments. We have a lot more designs, and so we are excited what comes out of this. As we are building upon all these building blocks and putting them together like graphs. So if we build using the Lego pieces to build protein interfaces, more complex than peptides. And what we are actually learning really is use the knowledge of proteins than natural proteins are to build new ones, right? Learning from the bird how to fly, but we don't need to replicate the bird, right? So we're just using the rules, the physical rules we are learning from our machine learning approaches to design new things that may be functional in ours. With this, I'd like to thank all the people in my group. This is Mike, who started all this in his graduation. Gianmarco did a lot of the work on T-cell receptor and development and binding affinity, and many of the other people here are now doing on different parts of the project on proteins and other things in the immune system that I did not talk. Thank you all for sitting through this. Happy to take any other question you have. Ed?
Right. So you used rotational invariance. Do you gain anything by including parity and time reversal?
Parity, I don't know. The time reversal, well, there's energy conservation, right? So, but there is
Reversal.
time reverse. Yeah.
Manipulation.
So we don't have any dynamical data here, right? So that would be a cool thing to have. That's something we are really interested in. Looking at molecular dynamics, right? But they're not that accurate. So one of the things we are interested in these days are enzymes. They change conformation. So the dynamics there is very interesting, and they're not reversible because the out of equilibrium process. Something we are thinking about. I think we're low on data on that side, but I think that would be cool.
Let me look at the question slightly differently. You have these Victor D functions. Only for integral angular momentum. But I think you could still get something. Well, it's a question. If you impose parity, would be another constraint.
Would be another constraint. Oh, I should repeat the question. Yes.
I have a question.
Okay.
Why if the peptide?
Yeah.
That's presumably for a specific T-cell receptor.
Yeah.
Well, what is the application you're using deciding the peptide? Usually the cells just going around looking.
Yeah. The question is, what's the application of designing a peptide? So there's a lot of hype. If some of you may have seen on cancer vaccines, right? There you are trying to promote a specific antigen because you know that there's a T-cell receptor going after it. The other thing is you want to identify a specific, you can design TCRs, right? So receptors. You want to know which peptide you want to target. So if you have a T cell receptor, you want to know if it is specific enough. You need to know the distribution of peptides that it could potentially bind to. That was the sort of motivation of this work. But in principle, you want to first of all know among the cancer antigens you have, you can sequence the tumor, right? You know which mutations you have, which ones are more pathogenic and can trigger an immune response. Then out of the T cells you get, which ones are more specific. That would be the application.
So this is kinetic proofreading, and selection is really on rates rather than affinities. How do you reconcile that with the last part of it, or do you really focus on affinities?
Yeah. So question was at the beginning of the talk, I talked about kinetic proofreading and how self non-self discrimination factors in kinetic proofreading. How do I reconcile? That's a kinetic question. And I'm looking at some binding affinity, which is like equilibrium proxy, basically. How do I do this, right? And the kinetic proofreading is basically trying to discriminate self and non-self. And that works very well in T cells. If you're a very good non-self, right, that's what we want to design. You're gonna mount a response, right? A kinetic proofreading is gonna even make your life easier. Really. That's that. So we are, I guess, overestimating the amount of self response from a T cell, right? But what we have seen from experiments is that the affinity or quality of antigen is actually a good proxy first step for identifying good candidates that elicit a response.
Yeah, so I have a technical question.
Yes.
So this seems like your method is very nice, but it does require knowing to some extent the structure.
Mm-hmm.
And in particular, in the finding case, you even have to know the structure, both the antigen and the antibody, right? Together. And then, so listen, there's another question related to that, which is that then you do that, and you use some rotational invariants. By themselves, yes. But together, are you sure that because you are mutating something in the middle?
Yes.
You think that can actually break that rotationalism?
Yes. Oh, so I'll repeat the question. Very good point. So model is good, it's generalizable, right? But it requires the structures as input, and we don't have the structures for everything. We have a lot more sequences. And and then say let's say we have the structure. Is the rotational equivariance a valid assumption when we are mutating at the interface of two proteins? So the answer to your first question, yeah, there's no free lunch, right? So you need a structure to make this work. We have done a lot of work using AlphaFold structures. It works very well if the structure is good. We have done actually on the TCR things, we've done AlphaFold free, so you can actually use the complex. You do it one by one because the code is not yet public. But in those cases, you can have really good designs starting from a computational structure. Now, about rotational equivariance, when you have, for most amino acid mutations, when you make one mutation, what we are doing is a local relaxation first, and then calculating things. Assumption of equivariance is that now I have a rotated version of this relaxed structure. It should be equivalent to the unrotated version, and that still holds. Right? So we're not looking at a rigid body around the amino acid. We relax it first and then calculate it. And the local relaxation in old-fashioned sort of Monte Carlo approaches are generally okay. So Rosetta and things like that are generally okay to move around the atoms, give you a room to put a new amino acid.
Yeah. And so actually this question looks right on, because at the beginning you were talking about how you're getting out. I was actually completely shocked that you said there were only 1,000 antibody structures.
of the order of thousands. So
Why is that? Like, there's a lot of, it seems like I wish I guessed
more,
a lot more, like 100,000, because
very important.
protein
Oh,
data banks.
yep.
Why is it so small?
So there's the question is, why do we have so few structures for antibodies? Even worse for T cell receptors, the hundreds.
After I saw the picture for the T cell receptors, now I understood that part. But
Antibody structures are equally difficult, and so the issue with this thing is experiment. So there is this very variable loop that is present for T cell receptors and antibodies, and experimentally, it's really a pain to get the structure of those things right. So T cell receptors, the main issue is the binding affinity is very low. And so you need to make a crystal structure, you need to stabilize this complex. That's really difficult. That's why we have only a few hundred. Antibodies, we have more, but then again, you have a disordered region in your protein that you don't get a good resolution when you build a structure out of it. So that's really a experimental difficulty. Now, the other problem with this is AlphaFold is a great tool, but no machine learning model generalizes beyond the things that it has seen, right? So I give a sequence of antibody to AlphaFold, it generally gives me completely nonsense structure, specifically in the region that I'm interested in, which is this disordered region. The only time that I get a reasonable structure is when I have something similar to that sequence already in the database, so then it can use that to refine it. So this is really the bottleneck of this method, and you're very much aware of it. So now we are actually, as the next one of the people in this picture, they're basically trying to do sequence structure model that is combined. So now you have two types of entry, and if you miss one, generally you're training on both data together. But that's what your model knows. But if you remove one part, you should be able to at least say something if you only have the sequence. So that's the direction we are going, because we think, forming sequence model with the structure is very useful. It's good to have a sequence model that works moderately well compared to a structure model that works perfectly well. The data.
Well, last time, could your modeling help improve the experimental?
I mean, not that I know. I've talked to experimental. I think it's really a technological step that needs to be worked on.
Last question.
So beyond the rotational equivariance for a single molecule, you said at the start that the immune system has this mapping from immune shape space to antigenic shape space. It's not entirely obvious to me how that mapping is preserved by this holographic CNN, and if similar shapes actually end up in the same space and clustering in your native space. Is there some way to show that?
It's something we're after. So the question is, I showed this beautiful sort of general shape space at the beginning. I went to looking at individual molecules, how do these things map, and whether things that have similar shape bind to the similar, what is shape, right? The definition of shape was things that are similar in shape, they bind to similar things. And so that's what we're after, right? Basically trying to now generalize from individual molecules using this model, like what kind of changes you can allow here to still bind to the same thing. So showed you the peptide design. We're designing T cell receptors, antibodies on the other side, right, for the same antigen. So that gives you the distribution of things you can vary that still target the same antigen. That gets at the shapes.
That's still local, right? Not the global.
If you design, you can sample globally, right? So I don't design starting from one initial and then deviate from that side.
Great. Let's thank Armita again for the